This lecture is about cell membrane. As you know, the cell membrane it follows a model known as fluid mosaic model. So this model, fluid mosaic model, was proposed by Singer and Nicholson that explains the components of cell membrane and its function. Okay, so there are two words, mainly the fluid and the mosaic. So fluid it means lipids, means that the lipids they provide fluidity to the membrane. We will see about both the structures further in the lecture. Okay, now the main points about it, that what are the components of a cell membrane. These are the carbohydrates, proteins and the lipids. So as you know that the proteins are the major component of the cell membrane. These are not the lipids. Lipids are only 40% present. Protein is the maximum component that makes up the cell membrane. So the other point we have is the inner mitochondrial membrane has highest concentration of protein present. It is a high yielding point. You can get it as an MCQ. Now we have the exception to this theory. Which theory? That the protein is the major component of the cell membrane. We have an exception to it. That is the myelin sheath of the nerve cell membrane. It contains lipid in more quantity than as compared to the proteins. Okay. Now the three things, the carbohydrate, protein and lipid. We will discuss three of them in this lecture. So firstly, the membrane lipids. Lipids are of three kinds that are present in the cell membrane. The first one is the phosphate group attached, the lipids that have the phosphate group attached. The second one is the carbohydrate group attached, and the third one is the sterols that are called as cholesterol. So the first one, the phosphate group, in the lipids that has a phosphate group attached in it, these are called as phospholipids. Then the carbohydrate group attached, that are called as glycolipids. And the third one, the sterols, these are the cholesterol, these are called as cholesterols. Okay, cholesterol, okay, they are called as cholesterol. Now the first one, okay, hope you are understanding that in lipids we have three kind of lipids. Okay, now we are taking this first group, phospholipids. We are taking this first group, phospholipids. Now in phospholipids also we have various kind of means phospholipids, like mainly we have the five ones. We will discuss each of them. The first one we are going to discuss is the phosphatidyl choline. It is also called as lecithin. It is most abundant phospholipid that is present. The most common site where we can see it is the pulmonary surfactant. It is useful for the assessment of fetal lung maturity. How we calculate it? It is the L by S ratio. L it is what, lecithin, and S is sphingomyelin. We will see about it later also. So both its ratio, L by S ratio, if it is equal or more than 2, then it is to calculate fetal maturity. It is used. It's okay. Like the concentration, it's good. Okay. Now the second one we have is the sphingomyelin. It is the lipid of the myelin sheath. As you know, for the conduction, fast conduction, we have lipids. So this is present in the sphingomyelin is present there. Also, it is present in the lung. It is a minor surfactant. Now, if we say the deficiency of sphingomyelinase, not sphingomyelin, that for used for metabolism, that enzyme. Okay, sphingomyelinase. It leads to a disorder known as Niemann-Pick's disease. Okay, so it has a characteristic of enlarged liver, enlarged spleen, and mental retardation. These are the these are the feature of this disease, Niemann-Pick's disease, and when it is caused when there is a deficiency of enzyme sphingomyelinase. Okay, this was the second phospholipid. Okay, now the third one, the third phospholipid, that is phosphatidylserine. It is present inside the cell. Why I'm telling inside the cell? Because these lipids are present either outside or inside. So this one is present inside the cell membrane, inside the cell. Earlier we have seen the phosphatidylcholine. It is present outside the cell. Sphingomyelin, that is also present outside the cell. This one's phosphatidylserine is present inside the cell. So it has a main feature like the exposure of phosphatidylserine. It is a signal. It means signal for apoptosis. Like when the cell has to go apoptosis, this protein would be outside, like it would signal that this cell has to be eaten. Eat me signal for apoptosis. Okay. Now the annexin A5 assay is used for detecting expressed this phosphatidylserine on the outer surface. Like this one assay is used for detecting apoptotic cells that are going to have apoptosis. It is used for that. Okay. Now the inside to outside, when it is expressed, as I said that it would be expressed outside. Normally it is present inside, but when the cell has to go for apoptosis, it would be expressed outside. So it is done by enzyme scramblase. Okay. Now the deficiency of scramblase would lead to a disorder known as Scott syndrome. Now the fourth lipid that we have is phosphatidylinositol. It is a precursor of second messenger of IP3 and DAG. The fifth we have is the cardiolipin. Fifth lipid we have. It is abundant in the inner mitochondrial membrane. Now two syndromes related to it are the enzyme involved in the metabolism of cardiolipin is tafazine. Okay, the gene encoding tafazine is mutated, then there would be a disorder known as Barth syndrome. Okay, hope you do understand that. That what I said, cardiolipin. Okay, now for its metabolism, this lipid its metabolism, an enzyme is involved known as tafazine. Okay, when this gene encoding tafazine is mutated, there would be a disorder known as Barth syndrome. Okay. Now, what would be the characteristic of Barth syndrome? These are the characteristics. Okay, it would be dilated cardiomyopathy, skeletal muscle weakness, growth retardation, and neutropenia. It is an X-linked recessive condition that affects only males. Now, the anticardiolipin antibodies. These are present in a serodiagnostic population test for syphilis, and this test is known as VDRL test. Okay, uh, these antibodies, anticardiolipin antibodies, these are present cardiolipin antibodies. These are present in a test in this zero diagnostic population test for syphilis. If this is present, the person is suffering from syphilis. Okay. Now, as I said, that lipids we have of three kinds. Phospholipids that we have discussed. Okay. Now we have the second lipids, that is the glycolipids. Okay, so we have glycolipids mainly of two types, that is the gangliosides and the cerebrosides. Two kinds, that is the cerebrosides and gangliosides. So this one gangliosides and cerebrosides, this name is only important, nothing much. Now GM1 ganglioside 1 acts as a receptor for microorganisms like C. For cholera toxin, it acts as a receptor and they act according to it. So we have as an example as cholera toxin and heat labile enterotoxin of E. coli. These both. The third one we have is cholesterol. So we have firstly like see we have like two kinds of fatty acids, that the unsaturated fatty acids and the saturated fatty acids. The unsaturated fatty acids, these are the good one. It increases membrane fluidity. These are the essential fatty acids: linoleic acid, linolenic acid, and arachidonic acid. These three are the essential fatty acids, means it increases fluidity. Whereas the second one we have is the saturated fatty acid. These are also called as trans fatty acid. It decreases the membrane fluidity. And the two examples we have is stearic acid and palmitic acid. These are not good for our health. These trans fatty acid as such, because they are fatty. These are saturated fatty acids, and what would they do? They would decrease the fluidity of cell membrane. Now, cholesterol is called as a fluidity buffer of the cell membrane. It maintains fluidity, nor it increases nor it decreases. It maintains. Okay. Now these essential fatty acids that I named, the linolenic acid, linolenic acid, and arachidonic acid. These are essential fatty acids. Now, as I said, that it increases it increases the fluidity. You might have heard about the omega 3 fatty acid. They are also good for our health, and it increases fluidity. They are also essential fatty acids. Now the second component of the cell membrane, that is protein. So proteins we have of three kinds: integral membrane proteins, peripheral proteins, and lipid anchored protein. So the first one, integral membrane proteins. These are also called as transmembrane proteins because they are present both inside and outside the cells. Their function is firstly that they act as a receptor, and these act as a receptor, and the second, they act as a transporters. Okay, for receptors, as an example, G coupled protein receptors. G protein coupled receptor, what it does, that it expands to the cell membrane 7 times. That's what also called as spintine protein. Okay, you will study about it later on. So it expands 7 times in a cell membrane. This one, G coupled protein receptor. The second one, the second function we have is as it acts as a transporter, as carriers or channel pumps. Example, sodium potassium pump. It acts as a carriers or pump. This was the first protein that we discussed, that is the integral membrane protein. The second one we have is the peripheral protein. So peripheral protein, they are supportive and give structural integrity to the cell. For an example, the shape of the RBC is due to alpha spectrin, beta spectrin, and chirin band 3 protein. These all give structure to the RBC. Now, mutation to any of these protein can lead to the change in the shape of the cell and thus the disorder. So, we have these are the mutated. Okay, spectrin mutation. Spectrin, when there's a mutation in it, what would it cause? It would cause hereditary leptocytosis. And chirin, we have a mutation, it would cause hereditary. Sorry, I was wrong. Spectrin mutation, it would cause hereditary elliptocytosis. And chirin, when there's a mutation in it, it would called hereditary spherocytosis. Dystrophin, its mutation called Duchenne muscular dystrophy. Okay, dystrophin it is also a protein that gives structure. It is present in the muscle, and it adds strength to the muscle. Third type of protein we have is the lipid anchored protein. Okay, so it is covalently linked to the glycosyl phosphatidylinositol. Now these both are the GPI of the RBC, CD55 and CD59. They prevent RBC from hemolysis, and when there is a deficiency, it would lead to paroxysmal nocturnal hemoglobinuria. Okay, when these both are deficient in a person. Now the last component we have is the carbohydrate, and as I said you that this is present in the least amount, the 5%, only 5% it is present in a human beings. Okay, thank you.